Greetings, Bill Mobley, Chair of the Department of Neurosciences, and bringing you the next edition of Neurosciences Connections. I have with me today a very important person in my life, Doris Trauner, who was interim chair of the department for a number of years and who has really distinguished herself as a UCSD faculty member in many ways, including her research on brain development and disorders of brain development. So, Doris, welcome. Glad you're here, and tell us about what you do and how you do it and what's going on now in your research.
My general area of research is on brain development and how the brain develops both in normal children and also in the face of some sort of early insult to the brain.
Mm-hmm.
So I study several groups of children in order to do this. I study children who had a very early perinatal stroke where they have damage to one area of the brain, and we can see that damage by looking at an MRI scan.
Yeah, so not everybody really understands this. Kids have strokes.
That's right. Kids have strokes, even babies have strokes.
Yeah. And I guess the causes of those strokes are quite different than adults. But what happens during a stroke in a young child or a brand newborn baby?
There are several things that can cause a stroke in a newborn. They can have a blood clot in an artery that goes to the brain, or they can have a bleed into the brain. Then the blood clots are caused by things like clotting abnormalities that either the mother can have or the baby can have.
Right.
Bleeding can be caused by disruption of normal clotting factors, so just the opposite of the babies who clot too much.
Right, right. So stroke's a real problem.
Yes.
And what you're saying is that depending upon how severe the stroke is and what part of the brain's involved, it can present some pretty huge clinical problems.
Well, that's the interesting thing about these children and why we're studying them, because it turns out that when an adult has a stroke, they have very predictable types of problems afterwards based on where the lesion is, where the abnormality is. When a baby has a stroke, that's not the case. And what we see is a great deal of the brain's ability to organize around an area of damage as the brain is developing.
So you're saying that strokes in children have different causes and also have different consequences than than strokes in adults?
Definitely, yes.
Doris, you've said that the brain is able to reorganize itself. Do we know much about how that happens?
No, we really don't, and that's what we're studying. We're using several techniques to try to figure out how the brain reorganizes after an insult like a stroke. And we're using neuroimaging techniques with MRI scans. We're using something called event-related brain potentials, where we look at the brain electrical activity during certain kinds of tasks, such as listening to words. And then we also do neuropsychological and behavioral tests, and we're hoping that by putting all of those together, and we can come up with a clearer idea of how the brain reorganizes when there's an insult like that.
You know, I guess one fantasy, we know the young brain is much more plastic than the older brain. I guess one fantasy is that by understanding the basic elements of reorganization in children and babies, we might actually help adults someday rewire their brains, at least recover some of the function that they've lost in the setting of stroke. Is that a potential outcome of this work?
Yes, there are many potential outcomes of the work. One is to figure out how to better help children who have some sort of brain damage early in life. Not only strokes, but from traumatic brain injury, for example. Many of these children suffer a lot of consequences, and we're hoping that we'll be able to figure out ways of helping them to improve their outcome. But it can also carry over to adults who have brain injuries like that. And the hope is that we'll be able to find some means of improving outcome for people of any age who suffer a brain injury.
Yeah, it's exciting, and it's obviously a hugely important topic with so many men and women in harm's way in the Middle East. This is obviously a very important topic for all of us. What's new in traumatic brain injury? Do we know more now than we did 10 years ago about the nature of the lesion and how it progresses?
We know some more about the nature of the lesion in traumatic brain injury, but there's still a lot that we don't know. And we're using some new and exciting neuroimaging techniques to try to learn more about the actual worst type of damage structurally that can happen with traumatic brain injury. So in the past, if you did a CT scan or an MRI scan, you might see an area of damage and you might not. But even when you couldn't see an area of damage, those individuals had significant functional impairments. And now with the newer techniques where we can actually look at the white matter integrity and look at much more detail, we see that, of course, the brain is not normal after a traumatic brain injury. And once we can define better what the abnormalities are structurally, then we can go on to look at, again, structure-function relationships and how specific kinds of injury lead to specific kinds of outcomes.
Good. What other disorders in kids are being studied by your lab?
We study a number of disorders. Another one is a metabolic genetic disorder called cystinosis, and this is a lysosomal storage disease where an amino acid accumulates in the lysosomes of cells and can't get out. And the neurological problem there, or the cognitive problem, is that these children have a specific cognitive profile of visual spatial and visual memory deficits on a background of normal intelligence and normal language skills. And
It's fascinating, isn't it?
Yes, it is. And we've been studying structure-function relationships there too, and looking at brain structure and how that differs in this group of individuals compared with people without cystinosis, and then how the brain structural differences affect cognitive functioning. And this has never been done before. In fact, it wasn't even recognized that cystinosis affected the brain until we got involved in this research a number of years ago and identified the cognitive deficits, and then more recently some specific structural abnormalities.
So when you find out, when you look at the structural basis and you discern that, and then you need to understand the functional connections that are disrupted, where does this go then over time? Do you envision that as you accumulate more information, you'll be treating kids with cystinosis in different ways, either behaviorally or otherwise?
Yes, there are at least two consequences, we hope, of the study. One is to be able to identify children very early who are going to have these deficits, because not 100% of children with cystinosis will have these visual spatial problems. So if we can find a biomarker for the disease that will be related to brain or cognitive outcome, then we can identify them very early and intervene early. Excuse me. And the second outcome we hope to achieve is to be able to intervene and find the most effective treatment for these children, and also to be able to monitor novel treatments for the disorder itself.
Using imaging, I guess.
Using imaging
and behavioral tests.
Yes.
Great. So that puts it all together in a way. It puts together the circuit and the behavior that emerges from the circuit, and it tries to diagnose it early, treat it effectively, but the imaging tools and the behavioral tools really let you say, is this working or not? Am I getting data from this treatment that really encourages me that it's doing some good?
That's right.
It's exciting stuff. How about the tools? Are the tools good enough for you right now? Are the imaging tools adequate to the task, or could you imagine even better tools in the future, and what would they be?
I think the imaging tools are very exciting. We could use even better tools, but what we're using right now are some techniques that are very exciting, called such as voxel-based lesion symptom mapping, where we use this with the children with strokes, where we look at where the lesion is and how the cognitive dysfunction maps onto the lesion in order to get at what parts of the brain are absolutely necessary for a certain specific function. So we choose, say, some expressive language measure, and we, using this technique, can identify what parts of the brain in neonatal life are necessary for that function to develop.
And is it clear now that even in neonatal life that there's pretty clear-cut differences, side to side, right versus left differences, even in those very early days?
Well, that's one of the really interesting things, is that there aren't a lot of differences between the right and the left hemispheres in terms of outcome, in terms of language development, for example, and intellectual development, when the damage occurs very early in life, very different from what happens with adults. But there are some subtle differences between the two hemispheres that are present very early in life.
And that's consistent from child to child?
Yes, that it seems to be. And using this voxel-based lesion symptom mapping technique, we actually find that some of the brain structures that are crucial for adult language are also crucial for language development in infants, even though the language outcome is better in the children than it is in the adults.
So in a way, this early work is even able to help you predict which brain regions might take over new functions, and then where, I guess, those folks who work at the level of individual cells and axons need to look to see how reorganization is occurring. Doris, you spend a lot of your time studying autism. Tell us where that field stands right now, what's new and exciting, and where's it going?
Well, there's a lot of research going on in autism around the country and in San Diego and at UCSD, and we have some specific studies that we're doing in my lab. We're looking at nonverbal communication skills in children with autism, and we're also looking at some higher level motor tasks or motor impairments in individuals with autism. We have some ideas that some of the core deficits in autism may actually be related to some specific motor deficits that have not been well studied in the past. So we've been focusing on those areas. Again,
so this goes beyond the stereotypy. We're talking about actual changes in motor performance.
Yes.
Now, this is not old news. This is a pretty new observation, it sounds like.
Well, there have been a few people who've been interested in dyspraxia in autism, but haven't really related it to some of the other deficits in autism. And I think that there may be a significant impact of dyspraxia on other developmental functions in these children.
How will this field develop going forward? What's your guess?
Autism is such a complicated set of conditions that I think one of the hindrances in the past has been the lack of subtyping of children with autism in order to try to understand more about the underlying pathophysiology. And so one of the areas that I think is most important is subtyping of autism, and then studying specific subgroups in order to try to understand more about what's causing the deficits, what's causing the autism in the first place, and then how to treat it.
And will the same tools be used? You'll use imaging tools, you'll use behavioral testing, that sort of thing will certainly be part of it.
Yes.
And where does the gene search fit into this whole thing? Presumably the subtypes might be more likely to give you a specific genetic lesion that one can pin down?
Yes, I think so. I think that in order to really get at the genetics, it's going to be very important to subtype these children.
Doris, thank you for being here. Very exciting work, really at the cutting edge, terribly important for understanding both normal brain development and also for the changes that occur in children with neurologic disease. Doris, thanks so much.
Thank you.